Spinal muscular atrophy is an autosomal recessive genetic disorder that is characterized by the degeneration and loss of motor neurons in the spinal cord, leading to muscle weakness, respiratory failure, and death. In fact, SMA is one of the most common genetic causes of death in children, with an incidence of 1 in 10,000 newborns. But to understand SMA, we need to first look at the relevant elements of RNA splicing. DNA is first transcribed to pre-mRNA, which contains non-coding sequences called introns and coding sequences called exons. RNA splicing, the process in which introns are removed and exons are spliced together, is an important process to form mature messenger RNA, which is then subsequently translated into protein. Splicing is achieved by the spliceosome, a protein complex comprised of 5 small nuclear ribonucleoproteins, or SNRPs. SNRPs recognize splice sites at the 5' and 3' end and an adenine branch point in the intron. The spliceosome assembles, and through the first transesterification reaction, a lariat is formed. Through a second transesterification reaction, the intron lariat is removed, leaving the 2 neighboring exons spliced together. Additionally, splicing is regulated by the exonic splicing enhancer and the exonic splicing suppressor sequences. ESE sequences recruit proteins that enhance splicing at neighboring splice sites, while ESS sequences recruit proteins that suppress splicing at neighboring splice sites, resulting in exon skipping. Mutations that cause aberrant RNA splicing often result in disease, such as spinal muscular atrophy. SMA is caused by a homozygous deletion or mutation of the survival motor neuron 1 gene, which codes for the SMN protein. The SMN protein plays a critical role in the cytoplasmic assembly of SNRPs into the spliceosome, as well as in pre-mRNA splicing, transcription, and metabolism of rRNA. The lack of SMN proteins result in widespread splicing defects of numerous other genes, which causes incorrect protein synthesis in motor neurons, leading to their atrophy. On chromosome 5, the human genome has 2 nearly identical SMN coding genes called SMN1 and SMN2. Compared to the telomeric SMN1 gene, the centromeric SMN2 gene has a single nucleotide variation from C to T in exon 7. SMN1 codes for the full-length SMN mRNA, which encodes for a fully functional SMN protein. The variation in SMN2 results in the exclusion of exon 7 in the majority of mature mRNA transcripts. The exon 7 excluded mRNA, shown here in black, encodes for a truncated SMN protein that is unstable and rapidly degraded, leaving only 10 to 20% of fully functional SMN proteins. In a healthy human being, SMN1 gene provides sufficient levels of functional SMN proteins to keep cellular activity normal. Problem arises when SM1 gene is deleted or mutated such that it no longer encodes for functional SMN proteins, such is the case in SMA patients. As a result, the only functional SMN proteins are the limited levels produced from the SMN2 gene. Cellular SMN levels are sufficient to prevent lethality, but not enough to compensate for the loss of SMN1. Here we have a mouse model of SMA. Motor neurons are labeled in green, and SNRPs are labeled in red. As SMN proteins are necessary for the assembly of SNRPs, we see that in mice with SMA, there is a lack of SNRPs in motor neuron nuclei. This results in splicing deficiencies of important genes necessary for motor neuron survival. How does the C to T transition result in the skipping of exon 7 in SMN2 mRNA transcripts? Although the C to T transition in exon 7 is translationally silent, it disrupts the exonic splicing enhancer in exon 7. A heptamer ESE motif lies in exon 7 of SMN1. However, the C to T transition disrupts and eliminates the ESE. Studies have found that the ESE in SMN1 exon 7 recruits SRSF1 proteins, shown here as the height of the black bar, while the disrupted ESE in SMN2 exon 7 does not. Normally, SRSF1, a splicing factor which facilitates splicing through a series of protein-protein and protein-RNA bridges, recruits U1 and U2AF, which in turn recruits U2 for splicing of exon 7. However, the C to T transition in SMN2 exon 7 prevents the efficient binding SRSF1, and this results in the skipping of exon 7. Currently, there are no treatments for SMA. However, knowing how exon 7 is excluded can help researchers find therapies that can increase exon 7 inclusion in SMN2 mRNA transcripts to ultimately produce functional SMN proteins.